Hello, everybody. So now that we know about the components of a cell, it's time to look at this most amazing part of the cell membrane, also known as the plasma membrane. Think of it as the gatekeeper for the cell. It determines what gets in, what can leave, and what cannot get in and what cannot leave. And in a sense, the cell membrane is the creator of the cytoplasm. So let's take a look at this. First of all, the basic component of the cell membrane is a molecule called a phospholipid. It's a molecule that is composed of some fats and a phosphate group. And I'll show you a picture in just a second. The lipid part is repelled by water, and the phosphate part is attracted by water. So as a result of this, phospholipids form a two-layered structure spontaneously. And what I mean by that, it doesn't require any additional energy. It just happens. Dump a bunch of phospholipids in water, and you get this bilayered or two-layered structure that we're going to see right now. Here is the phospholipid bilayer, as it's called. And you can see these gray ovals are the phosphates, and the yellow tails are the fats, the fatty acids. And so if you stick these molecules into water, the phosphates all line up on the outside facing the water, and the lipids, the fatty acids, are all on the inside. And this is the basic structure of the cell membrane. Here is a more detailed look at the phospholipids. Here we have a schematic representation of it. Here we have a representation of the actual atoms that are in it. And then here is a structural formula, as it's called. And what I want you to notice is that there's a 3-carbon chain in here called a glycerol. And on one of the carbons sits the phosphate. And on the other 2 carbons sit the fatty acid tails. The phosphate attracts water, and the fatty acid tails repel water. The outer surface is therefore called hydrophilic. In other words, it loves water, while the inner surface is hydrophobic or hates water. And the water molecules therefore cluster around the phosphate end and physically turn the molecules so that the hydrophobic ends are pointing towards each other, and the water is what creates this phospholipid bilayer, which is the basic structure of the cell membrane. Now, the membrane is fluid, and the individual phospholipids move around the membrane. And the membranes have to be fluid, not solid, not frozen, to work properly. And they're about as fluid as salad oil. If you just had a bottle of phospholipids, it would feel like salad oil in your hands. So here's how it looks. You can see the water molecules here clustering around this phosphate end, this hydrophilic end, and pointing the hydrophobic tails towards each other. And then here is what I meant about the movement. These phospholipids change place with each other about 10 million times per second, so they're in constant motion. And they flip across the membrane about once a month, so that kind of movement is much less common. And that's what we mean by the membranes being fluid. Things will move around in the membrane. Now, as temperatures cool, membranes switch from a fluid state to a solid state. In other words, they crystallize, just like you get water cold enough and it forms ice. It goes from a liquid state to a solid state. The temperature at which a membrane solidifies can be changed by different types of fatty acids in the tails of the phospholipid. So you've probably heard of unsaturated fats. Well, unsaturated fats have a bend or a kink in the tail, and as a result, they resist freezing and don't freeze until a lower temperature. Let's take a look at this. So here we can see an unsaturated fat with a kink in the tail. This, on the other hand, would be a saturated fat because it would not have a kink in the tail. So a saturated fat is straight, and as a result, they solidifies or freeze at a higher temperature. Now give yourself a second to embrace that idea. You put the unsaturated fats into a phospholipid, and it lowers the freezing point. If there are saturated fats, then the freezing point is higher. Here we have a picture of this. Here is a membrane that has lots of unsaturated fats in the tails, and therefore it's a very fluid membrane. A membrane like this is much more viscous and it will freeze at a higher temperature. So here's a concept check. I'm not going to give you the answer today, but perhaps you can come to class with a question if you don't understand. Why do the plasma membranes in warm-blooded mammals contain a much higher percentage of saturated fatty acids than the cell membranes of organisms that can't control their temperature, like plants such as olives or soy or canola, which have a much higher percentage of unsaturated fats? So see if you can figure that out. Here's what we know about membranes. Membranes must be fluid to function. That's important that you remember that idea. At low temperatures, the membranes will solidify and not be able to function. But what about high temperatures? What will high temperatures do? Well, as we're going to find out in the next lecture, when you increase temperatures, it causes molecules to move more rapidly. And so a higher temperature will mean the molecules, the phospholipid molecules in the membrane, will jiggle faster. And therefore, the membrane will be more likely to become unstable and lose its contents or burst, thereby killing the cell. So membranes can only function within a relatively small range of temperatures. Cholesterol, which you all have heard about because of its health repercussions, is a molecule that is a normal component of the cell membrane. So what the heck is it doing there? Well, it turns out that at high temperature, cholesterol, which is pictured here in the upper right, reduces the movements of phospholipids and therefore keeps the membrane functioning at higher temperatures. And at low temperatures, it prevents solidification or freezing and therefore keeps the membrane functioning at low temperatures as well. So we can say it acts to reduce the effect of temperature, and therefore it acts like a buffer. A buffer is a substance that reduces the effect of something else, in this case temperature. It is a temperature buffer. Now, another thing about membranes is if we just empty their contents and then analyze them chemically, we find out that there is a lot of protein in the cell membrane. So what's up with the proteins? What are they doing there? Now, before we go on, let me tell you a little bit about proteins. Proteins are chains of smaller components called amino acids. So here are some free amino acids, and if you put them into a chain, you get what's called a peptide. And then if you fold that peptide up, you get what's called a protein. So the amino acids can be likened to a string of beads on a necklace. And there are about 20 kinds of amino acids that we use in our proteins. These molecules are large, and so they are called macromolecules. The macromolecule is protein. The building blocks are amino acids. So here's a concept check. What are proteins made of? 3, 2, 1. Got it? Are proteins large or small? 3, 2, 1. I hope you have that answer. What word that rhymes with pain might be used to describe the way amino acids are strung together in a protein? See if you can get that one. What word is used to describe large biological molecules? So what are these membrane proteins doing? Well, one thing they can do is transport substances across the membrane, and we'll be talking more about that in the upcoming days. They can also carry out chemical reactions at the surface of the membrane. And when a protein carries out a chemical reaction, it is called an enzyme. It could transmit signals from the outside to the inside and vice versa. It could join cells together or attach them to support structures. And it could recognize other cells nearby. So these proteins have a lot of different functions, and we're going to find out more about some of them as we continue in the course. Here's our picture now of what a membrane looks like. We have the phospholipid bilayer here, and then embedded in it are these proteins. And since the membrane is liquid, the proteins are able to move around in the membrane. This model of the membrane is called the fluid mosaic model. And what I would like you to do after you've watched this video is watch these two videos, which both give you some idea of what a membrane might look like. So that's it for today. I hope what you've come to understand is the phospholipid bilayer that forms the basic structure of a membrane, And embedded within the phospholipid bilayer are proteins. That these proteins have numerous functions in the membrane, including transport of materials across the membrane, carrying out chemical reactions, transmitting signals, recognition of other nearby cells, attaching cells to each other. In tomorrow's talk, we are going to look more at the process by which water flows across membranes and what the consequences of that are. And we will come to understand the process of osmosis. Until then, go outside, look at nature, ask some biological questions, and I'll see you soon.